Can you hear me? Welcome to this seminar session, which is part of Life Sciences seminar series here at Masaryk University in Brno, Czech Republic. My name is Ctirad Hofer, and it's my great honor to introduce our guest speaker today, who is Jayakrishnan Nandakumar. He is expert on structural biology of telomeres, and he is a very good friend of mine. As we spent several enjoyable months in laboratory of Thomas Czech in Colorado University in Boulder, I know him a bit, so I'm going to introduce him based on my experience and real-life stories. So when we first met, I immediately realized that Jayakrishnan is a really bright person, thanks to his clever mind and generosity. So this is something that you are going to enjoy today a lot. Then we started to collaborate at the laboratory, at the bench, and I shortly find that he's very skillful, especially when he broke his arm, and he could do all the laboratory work and also writing with his left hand. He's right-handed. So maybe you would like to see us in that time. Here we go. So you can see that we both are the same. The only difference is the amount of my hair. So this picture is symbolic because it was taken in the time when Jayakrishnan was leaving the laboratory of Thomas Czech after several successful years, decorated by really tremendous findings and exceptional publications. And if you look at the picture at the back, there is a clock, which is symbol our great time that we spent together back in Colorado. The clock is also symbol of new beginning because it was one of the first items that Jayakrishnan had in his independent laboratory at the University of Michigan. So the third symbolism connected with clock is connected with the topic of this seminar, as telomeres are molecular clock that serve as counting of lifespan of the cell. So please now join me in welcoming Jayakrishnan Nandakumar to Masaryk University by applause.
I'm audible, right? Okay. So maybe I'll start by saying that Ctirad is one of probably 10 non-Indian people who has the courage and boldness to pronounce my full name. And he does it perfectly. And usually that's why I have this on the title slide, because JK is a lot easier, but he pronounces my name perfectly. Okay, before I begin, I really, from the bottom of my heart, want to thank Ctirad for being such a wonderful host. I was actually at his house yesterday. He's hosting me even, at his home in addition to at his institute. And I was joking to him and his wife that he really shouldn't be having guests over because they might just stay there and not leave, because he's just so nice and such a great host. I also got a chance this morning to tour some of the facilities here, and they're just amazing. So I think I went to the cryo-EM facility. It's, top of the notch facility. I wish, I had access to something like that where I am. and then also the NMR facility. I don't do NMR, but just looking at all the infrastructure here, it's really for all the students in this audience. You have everything here that you need to be successful. All you need to be is motivated and hardworking. Again, just gauging by the people here, I think most of you, many of you are students, trainees of some kind, and so don't feel like if this is not your field of expertise, and that might be true for 90% of you, that you have to switch off or like, oh my gosh, I don't even know this. I know equally less about your field. Okay, so don't be shy to ask questions. There is no such thing as a bad question. You can interrupt me anytime, or you can wait till the end. It's up to you, but I have a tendency to just go on, but if you just wave or shout out, I can stop and answer your question at any time. Okay, so with that, maybe we get on to business. And as the name suggests, I'm going to talk to you about telomeres, but probably not so much about at least one of the major roles that, that telomeres perform in the cell. And I'm coming from University of Michigan, which is kind of, the state of Michigan is shaped like, a mitten, a hand, and it's in the Midwest of the United States. Okay, so this is a very, very, very brief introduction on telomeres. What it's showing you is they're at the ends of chromosomes. What is not shown here is the actual DNA sequence. It's a repetitive sequence. It's GGTTAG times thousands in humans and other mammals, for example, right? And so those are kind of tagging or coding the ends of your chromosomes so that this complex called shelterin, 6 proteins, can effectively bind and protect the ends. Why is this important? This is important because these DNA ends, or our natural chromosome ends, look like DNA breaks. And the last thing you want is your cell to fuse one chromosome to the next, right? So a major function of shelterin, or telomeres in general, is to protect these ends, right, from the cell's own DNA damage response and repair machinery. Okay, so why is it so important? What happens if you knock out one of these proteins? If you knock out one of these proteins, instead of having nice, pretty chromosomes colored in red with telomeres in this green color, you will end up with these trains, literally, of chromosomes fused to chromosomes, fused to chromosomes. Okay, so that's how important the shelterin complex is. This is its primary function. However, like I was saying, our lab has kind of branched into other areas of telomere function that are important, but are not directly related to the end protection function. So one of them is this regulation of telomerase, and we'll get into what that is. It's the enzyme that extends these ends, and is kind of related to the clock that one of the clocks that Sirad was referring to of cell division. And so what I'll do there is, it's all published data, so it's nothing new, but what I wanted to give you is kind of a 10-year update, the time since I knew you first met Sirad to today, what we've done and what the field has achieved as a whole. And then in the second half, touch upon another function, which I really feel this is the best place in the world to talk about it, which is the role of telomeres, a specific role of telomeres in the cell division process called meiosis. Okay? And so for the first part of the talk, what is important is this clock Sirad was talking about. DNA polymerases are not able to fully replicate the ends of chromosomes, and so every time a cell divides, its DNA gets shorter and shorter and shorter, and then ultimately a threshold is reached where the cell stops to divide and enters senescence, right? And so this is really an end replication problem, but is actually a good problem to have for 99.9% of our cells, because it serves as an anti-tumorigenic mechanism, right? It prevents cells from just going rogue and multiplying forever, right? However, if you are a stem cell, then you really need the capacity to divide over the entire lifetime of that organism or species, right? And so how do you solve the end replication problem? That's where telomerase comes in. I really should update this cartoon, because now there are high-resolution structures of telomerase. This was literally from the time I was a postdoc, where that was our understanding of telomerase structure. So there is a protein subunit TERT that I depict as a blue cartoon or a blue oval, and then an RNA subunit that contains a template sequence that helps telomerase synthesize telomeric repeats, right? And it doesn't stop at one. It can kind of realign and synthesize multiple repeats. And so what you can see is that it can kind of tend to compensate for the telomere shortening that happens because of the end replication problem. Okay, so this is obviously a good thing to have in stem cells, but it is also unfortunate that although telomerase is absent in normal somatic cells, right, non-stem somatic cells, it is activated in at least 90% of all cancers, irrespective of type, kind, etc. Right? So it is a major, at least on paper, a major target for anti-cancer drug design. Okay, but the flip side, telomerase is also a good cop, like I said, because you need it in stem cells, right? And so what happens if you have mutations in genes that are important for telomerase function, right? And so then what happens is you end up with these very unfortunate stem cell dysfunction diseases, the most prominent of which is, I would just call it DC, just for sake of remembering for the rest of this talk. And there what you have is it's diagnosed at the level of skin lesions, telling you that there's something wrong going on with the skin stem cells. They're not able to properly renew. But then the primary cause of death is bone marrow failure that basically comes from the hematopoietic stem cells, the blood-forming stem cells, not able to keep renewing throughout life. And it really is a telomere shortening disease because every patient thus far diagnosed with this disease has very short telomeres, sometimes below the 1 percentile of the population. And for at least 70% of the cases, it's a 1 in a million disease, so it's rare. But in 70% of the cases where it has been diagnosed, the mutation can be mapped to either telomerase or some telomere-related gene. So it really is a telomere dysfunction disease. Okay, so this is kind of a general background for telomerase, what happens if you don't have proper telomerase, and then zooming in completely to what I was interested in as a postdoc, right? My question was, if shelterin is so good at protecting these ends, how then can telomerase extend these ends? How can shelterin protect chromosome ends from everything, but allow telomerase to access these ends, right? And so the answer lies in this protein TPP1 that not only protects chromosome ends by virtue of being part of the shelterin complex, but then also by binding and recruiting telomerase there in the S phase so that telomerase can extend chromosome ends, okay? And so the big question I was asking is, again, this is 2009, 10, 11. It's all a haze now. Is, how does this protein A bind protein B? As simple as that. We had no structure of telomerase. What we did have is the structure of the domain of TPP1. It's called a TPP1 OB domain. Okay, it's a small domain and structure was solved. And so the simple question I asked is, can we find the surface of TPP1 that binds telomerase? And the way we went about asking it is as simple. The residues that bind telomerase will be A, on the surface of the protein, right? And B would be conserved because it's an important function. So based on that, we did, made a bunch of mutations on 16 different amino acids. And what we did is put those mutants, it's like a mutagenesis screen, but a directed one, right? Through 4 assays, and I won't show you each of those assays, but imagine there is a way to measure binding of TPP1 and telomerase. Imagine there is a way to recruit telomerase to telomeres in a living cell, right? Imagine there is, telomerase is an enzyme, it has an enzymatic activity, and I'll show you examples of these in later slides. And then finally, imagine we can measure telomere length. This is done with southern blots, and you would basically see telomeres as smears, right? So using these assays, we could show that there are 7 residues, and we call this bunch, this surface patch, as the TEL patch because it's TPP1's glutamate, so E is glutamate, and leucine-rich patch, okay, as the region that TPP1 uses to bind telomerase. And that really was the extent of our understanding of how telomerase is recruited to telomeres when I was a postdoc. And then, that's when I met Stira, then we had a paper, I got a job, I moved to Michigan. And then we learned from our collaborator, who's actually a doctor who specializes in studying DC, that disease that I talked to you about. And she met with a patient, 5-year-old boy, who actually has a mutation, a deletion of a lysine in TPP1. TPP1 is a long protein. The OB domain is one of its domains. This mutation is in that domain, okay? And clearly, that boy obviously has very short telomeres compared to what is average for his age. So this is just showing you a percentile, so this is where the 50th percentile is, and he's way below 1 percentile, right? Really short telomeres, has disease, and her question was, why, right? And it was quite obvious when you look at where that lysine is compared to the tel patch that I had identified in my postdoc, right? So it really smells like this is affecting recruitment. However, we are structural biologists, partly at least, and what was odd to us is when you look at the structure, all those glutamates of the tel patch are pointing out, meaning they want to bind telomerase. This lysine actually was pointing into the core of the protein. So why should deletion of that lysine result in a phenotype? So what we hypothesized is, well, it's not really having a lysine there, but having something there. Because if you delete the entire residue, you might now completely disrupt the folding of the remainder of the tel patch. Okay, so we hypothesize an alanine mutant is going to be fine, a deletion mutant is the disease mutant is going to be defective, and then these are the assays that I was talking to you about. And again, the people who did the work are shown with their names and pictures on the right. So this is a telomerase processivity assay. This is really cool. You have a primer, you have telomerase, you put in hot nucleotides, and remember, each telomeric repeat is 6 nucleotides. You basically see a ladder that goes up, and each rung is 6 nucleotides in length, right? So telomerase alone has this profile. If you add in TPP1, this was long known even before I started working on telomeres, is that because it recruits telomerase, that telomerase becomes more processive, meaning the telomerase sits on a primer and keeps synthesizing DNA so that you see preferentially longer repeats as opposed to shorter repeats. And then the question is, what happens when you make the disease mutation? You see that profile kind of shifts down. It's not very processive. The alanine mutation, like we guessed, is processive. And again, the quantitation is on the right. And then, of course, the EE to AA is like the tel patch mutant that I had identified in my postdoc. It's like a positive control for the mutation, where obviously you lose processivity. Again, clearly showing that the disease mutant is defective, and that the reason it's defective is not because that lysine side chain is important, but you just need some residue in there, right? Again, this is the other assay, which is telomerase recruitment. You're looking at a HeLa cell nucleus. The green dots you see are where TPP1 is sitting, meaning, it's the telomere. The red dots are basically, or puncta, are telomerase RNA, so meaning telomerase. This is using fish. And so the idea is, if telomerase is at telomeres, when you merge these images, you should see yellow, which is what you see for the wild type, but you clearly see how you have a defect with the disease mutation, but not so much with the alanine mutation again. And then to actually show that indeed the deletion of the lysine is disrupting the tel patch loop, we solved structures. So the wild type structure was already solved. That's the one in green by Ming Lei when he was in Tom Czech's lab. We solved the structures of the 2 mutations we were studying, or the mutants we were studying. And clearly, I hope you can see, zooming in on that loop, you basically displace those tel patch loop residues when you delete the lysine, but not when you mutate it to alanine, right? And so the final point here, this is very important for these diseases, is correlation versus causation, right? It's always a question. Just because you find a mutation of your favorite gene in a disease doesn't mean that mutation is the sole cause of that disease, right? I mean, we didn't go to the extent of proving it's the sole cause of the disease, but at least we asked, is this mutation sufficient to shorten telomeres in some other random cell line where you have wild-type TPP1. And so that random cell line happened to be HEK293T cells, which are easy to grow. We used CRISPR, which was just up and coming at that time, for knock-in. Of course, knockouts were more common. And the strategy we used is, well, we cut with guide RNAs at these positions, and then we provide a template that lacks that lysine codon. And then what we had is like a control where we still do the CRISPR, but we provide the wild-type template. How do you know that you edited the cells? We also introduced these silent mutations, right, that don't change the coding, but then we can later do PCR and restriction digestion to say, okay, this was CRISPR-engineered clone, either wild type with an asterisk or mutant. And lo and behold, this is a southern blot, again looking for telomere length. The smear moving up means long telomeres. The x-axis, if you may, or the lanes going from left to right are the time, days in culture. And you clearly see that cells that have the mutation actually do show telomere shrinking, clearly showing that this mutation is involved in telomere shortening. Okay, so that was kind of the beginning to the end of the TEL patch. So we identified it through this manual mutagenesis screen, but there's an actual human, at least 2 humans or 2 families that we know of that have a mutation in that protein that reduces telomere length, results in disease. So is this it? Is the TEL patch the only thing that binds telomerase? Right? So again, in our lab, this was through serendipity. What we observed is there's a second region, the N-terminus of the OB or the knob, that is also important for binding telomerase. Okay, how did we surmise this is so? When the TPP1 OB crystal structure was solved, long back, right, 2009, it actually came out as a dimer in the crystal, right? And how does it form a dimer in the crystal? If you think this is the OB domain, my palm, and this is the N-terminus, you basically had the N-termini from 2 different protomers coming and forming a dimer. But we know that TPP1 is not a dimer in solution. It's a monomer, right? So what we guessed is maybe that N-terminus wants to bind telomerase or something, right? And since you don't have any of that in your crystal conditions, it just kind of self-satisfies by forming this dimer, right? So maybe it's important. So how do you test it's important? We do the same assays, right? Mutate it, do the same assays that I just showed you, telomerase processivity assay, whether you delete the knob or make alanine mutations, you reduce telomerase processivity, you reduce telomerase recruitment to telomeres. Again, just to quickly focus, the telomerase foci in these mutants are not coinciding with the green telomere foci. And we did the telomere length analysis. This, of course, was not CRISPR. We overexpressed either wild type or mutant, and clearly overexpressing wild type actually increases telomere length because you have more TPP1 and brings in more telomerase, we think, whereas the mutant actually still decreases TPP1. Okay, so at the end of all this, around 2009, 2010, we kind of defined that TPP1 uses 2 regions. One is the TEL patch from my postdoc days. The other is the knob to actually bind telomerase, right? But really the big question then became, how does telomerase bind TPP1? Because more people know of telomerase than of TPP1, right? And it was difficult to answer because telomerase is a beast. It's a huge complex of several proteins and an RNA subunit, and how do you figure out where it binds? Right? And so to do that, what we got inspiration at that time, there was no human telomerase structure, but there were a bunch of tetrahymena. Tetrahymena is a ciliate. It's a single-celled eukaryote where many seminal discoveries have been made in many fields, including telomeres. Okay? And so its telomerase structure was solved, and instead of looking at this entire structure, which is complicated even for me, if you just focus on the green, the purple, and the cyan, what is shown are the green and the purple are the 10 and the IFD domains of the TERT, the protein subunit of telomerase, the catalytic protein subunit. And what you see is that in the primary structure, the 10 and the IFD are far apart, right? But when you fold this into a complex, you see how they form an interaction. Even more interesting is that there is a third protein that is forming this 3-way interaction. So the 10, the IFD from telomerase, and then this P50, which is basically a TPP1 homolog in the ciliate, right? So we were like, okay, we don't need to mutate the whole telomerase holoenzyme, because then the student who I suggest this experiment to is going to quit the lab and run away, right? 450 kilodalton complex. Instead, maybe all you need to focus on are these 10 and IFDs, again, if this whole structural solution is conserved in humans. But that is also a guess, right? How do I tell a student, just mutate 10 and IFD, we'll find something, and I'm sure it's going to work, right? And so what we did, exploited, to first answer the question, is it the 10 and IFD in human telomerase? We know it's in tetrahymena telomerase. That is important for binding TPP1. What we basically exploit is the fact that human telomerase can stimulate human telomerase is stimulated by human TPP1, but not really mouse TPP1, and vice versa with the mouse telomerase, suggesting there is a species specificity, right? A certain telomerase works only with its cognate one. Okay, how did we exploit that? Okay, so this is just a control lane. You have human telomerase, it has some activity, lane 1. You add in human, H for human, TPP1. I hope you can see how the pattern changes from lower running bands are intense to higher running bands are intense, right? If you give it the human telomerase, the mouse TPP1, not so great, right? So this is the species specificity. Now do the opposite species specificity. You have mouse telomerase, and if you don't add in any TPP1, it has some activity and processivity. If you add in the human TPP1, it does stimulate some. I hope you can see the difference here. But see how it goes like gangbusters if you give it its cognate TPP1, right? Compare this lane to this lane, right? So it really likes the mouse protein. So the question we asked is simple. Instead of giving it the full pure mouse tert, can we switch and put in just the human 10 domain into a mouse tert, or the human 10 domain and the human IFD into the mouse tert, and make that protein work with a human TPP1 protein? Is that clear? You just change those two domains into human. Now, does it work with a human TPP1 protein? And that's exactly what happened. You basically, I will skip to the final one, which is you have mouse tert, but we switched its 10 and IFD with the human versions. And now it actually is stimulated more by human TPP1, the middle lane, than it is by the mouse TPP1. Okay, again, this is just quantitation showing that. But basically what this shows is that as long as you have the mouse, or as long as you have the 10 and the IFD domains, that's all you need to get interaction with the TPP1. Okay, so then with that confidence, we did a mutagenesis screen, and it was done by my graduate student who did successfully complete his thesis before leaving the lab and publish papers. And basically, it's just the same routine of the way we did it for the tel patch, but we now made mutations either in the 10 domain, found many mutants. Again, I'll spare you the details. We did the same thing in this IFD. It's also called the IFD trap. That is the language that Julie Fagan, the person who solved the tetrahymena structure, uses. That also, there also we found mutants. And basically then what we did is, since the human complex, again, remember this is 2019, is so similar to the tetrahymena complex in terms of function, we created a structural homology model, not of the entire telomerase, but just the parts that we care for, right? The 10 domain, the trap, the TPP1, and then we highlighted residues that we have discovered from our screens, right? And you clearly see all those residues are kind of at the interface of what we think is telomerase TPP1 interface. Okay, so that's where we stopped. And where is the field currently? So this year, in the last 3 months, there was one paper from Kelly Nguyen's lab in MRC, in Science, and then Julie Fagan's lab in UCLA, both of which captured the human telomerase with TPP1 using cryo-EM. Again, showing you the power of cryo-EM, right? And what I want to show you again is, I hope you can see similarity between the two views shown here, is that what we had it was a best guess from our homology model in 2019, where you have the knob and the tel patch binding the 10 and the IFD domains. The actual structural interface of the human complex is indeed very, very similar to that of the tetrahymena complex. Okay, again, this one is from Kelly's paper, Kelly Nguyen's paper. All right, so that is kind of the end of part 1. Did we want to take questions? We can take questions now, or you can think about it and crystallize questions and ask, no pun intended, ask at the end. But any questions? Or I can carry on. Okay, we can carry on. And again, you should feel free to interrupt me. Okay, so the second part of my talk, like I said, has to do with meiosis. And I honestly didn't even know about this function of telomeres because I was trained as a chemist who became somehow a biochemist, structural biologist, and took my last meiosis class in sometime in high school or something. But we kind of stumbled into a structure that happened to be of a meiotic complex. And so that's when I started reading these reviews and understanding how amazingly beautiful genetics is, of course, and I don't have to tell that to this audience, but also how it is also true about meiosis. So meiosis is a specialized form of cell division. Light doesn't happen everywhere in your body, only in germ cells. It's one round of DNA replication followed by 2 rounds of cell division so that you end up with haploid gametes, right? 1n chromosomes. And an extremely important step, right? An extremely important step that actually kind of forms the basis for Mendel's primary observation of segregation of chromosomes is the homologous recombination that happens in prophase 1. So it happens in meiosis 1, specifically prophase 1, where all homologous chromosomes, so the dad and mom chromosomes, kind of recombine with each other. This is important for the segregation of chromosomes later on in meiosis, but also important for increasing genetic diversity, right? And so how does that happen? How does this not happen in any other cell at any other time, but happen in germ cells, right? And so it happens because in prophase 1, telomeres don't stay just inside the nucleus. They go and attach to the nuclear periphery. So the nuclear periphery is basically the nuclear inner and outer nuclear membranes, right? The telomeres attach there, and then they start moving. There is this transient bouquet complex where they're all close to each other, and then they basically come out finding their pair. Right? So the movements help the homologous chromosomes to find their homolog, at least that's the thinking, and then they pair up, undergo homologous recombination, and then everything falls apart. Basically, the telomeres retract back into the nucleus, and things look normal once again. Right? And so the question here is, how do these chromosomes move and pair in meiosis 1? And the answer lies in dynein, which is a motor that walks on microtubule tracks. Okay, so the microtubule track is shown as these gray lines, and we're trying to show like a hard-working dynein, like carrying cargo. Sitting in the cytosol, separated from its cargo by 2 membranes, right? And so how is dynein really doing that? That is really a big question, I think, in meiosis, because the nuclear envelope is intact when it is moving these chromosomes. But imagine that it can, and it does. You then get pairing of chromosomes, you get homologous recombination. Okay? All right, so the question is, how do you connect something in the cytosol to something in the nucleus without breaking the nuclear barrier? You do that, again, if you focus in on the nuclear envelope, using this complex of proteins called sun cache proteins. Okay, the sun protein kind of is a nuclear protein that spans the inner nuclear membrane. The cache protein is also a nuclear envelope protein, but it's on the outer nuclear membrane, and then it extends into the cytoplasm, and in the intermembrane space, on the periplasmic space, they kind of bind each other, okay? The sun1 and the cache5, right? So they kind of form the glue that connects dynein to telomeres. So that at least tells you how they're connected. But the question really is, how is dynein activated? And again, I have not talked at all about dynein. So what do I mean by even activating dynein? It's a motor, it should be switched on, right? Turns out you don't want your motor to just run if it's not carrying cargo, right? So there has to be a mechanism where you only want dynein motors to run when they're holding their cargo, so that you're not just wasting ATP, right? So how is that activation done? And so this is how dynein looks like. So this is its motor domain. These are the legs it uses to bind the microtubules and walk on it. And then, so this is the heavy chain, but it has many other chains, including this LIC, okay, that I want you to kind of remember because it's important. It's the light intermediate chain. All right, so it has that. However, it does not look like this when it is not attached to cargo. It looks more like this, auto-inhibited. Again, this is from structural data. Okay, so its legs are crossed and it cannot walk if it's not activated. So the whole activation process is to take dynein from the left-hand side to the right-hand side, and that happens with 2 factors, or complexes, I should say. One is this humongous dynactin complex. So I used to think telomerase is big, but then dynein and dynactin are like in a different scale bigger. Okay, but once that dynactin binds, and then the most important protein here, And this is different for different biological contexts. An activating adapter, literally adapter, it connects dynein to the cargo. When both of those bind, now dynein starts running on microtubules. Right? So the question really, in a nutshell for us is, when we say how is dynein activated in meiosis, we are asking what is this brown activating adapter for dynein during meiosis, right? So that is kind of, and then the really cool part is you can actually look at dynein running, in vitro using TIRF. Okay, single molecule TIRF, total internal reflection fluorescence microscopy, and basically if you fluorophore-label the dynein, you can actually see it start moving, okay, on microtubules. Of course, the microtubules are not imaged in this, but if you do image the microtubules, you will see that the dynein spots, the single particles are actually moving on dynein. So it's really easy to assay. Easy meaning feasible. Not easy for me. All right. Okay, so that's good. And so now more about activating adapters, right? Before we start hypothesizing what our activating adapter is, we need to know what they look like in general, right? So activating adapters, as the name suggests, has a big part that binds dynein and a part that binds the cargo. They need to connect the two. More specifically, how do they bind dynein? You see here, the end domain of an activating adapter binds this LIC, light intermediate chain of dynein, doesn't bind it anywhere. There is a specific helix at the C-terminus that has this phenylalanine motif, and known activating adapters actually have been crystallized with this helical peptide and shown to bind it. Okay, so that's how it binds. And then there is a central domain that, yes, binds dynein, but is also important for forming this coil-coil dimer of the activating adapter. Okay, and then like I said, the C-terminal domain doesn't bind dynein, it binds the cargo. Okay, so this is how it should look like, right? And what are the different families of activating adapters? Well, they're just separated into families based on what their end domain looks like. The rest is pretty similar in the sense, functionally, this would be a dimerization domain. This would bind cargo. Again, cargos can be a whole Golgi, can be something else in the neuron, mRNA. It could be all different cargo in the cell, several different cargo in the cell. Okay, and then there are three families: EF hand, hook, and CC, based on this LIC binding domain. Okay, so the question really is, do we have something in meiosis that looks like one of these three and has this domain arrangement and functions, right? And so this is really the work of one student, and it's unpublished. It's Ritvija Agarwal, and basically her work, like I said, is in my lab doing a lot of the biophysics and biochemistry, but I don't know to do TIRF. And so for those experiments, she worked in another lab in the same department, Morgan DeSantis' lab, and then we worked with our longtime collaborator to kind of validate our work in vivo, okay, in mice. Okay, so coming back to that picture I showed you, there's sun cache, and this question mark here is based on previous studies that have shown, using co-immunoprecipitation, so not purified proteins, that Cache5 associates with dynein components, right? So that was our clear hypothesis. Well, Cache5 binds dynein, and so it is the activating adapter, because on the other end, Cache5 is bound to the cargo, right? Ultimately chromosomes. So it makes sense. If you look at the domain diagram, it also makes sense. The N-terminus is an EF hand. We know EF hands are found in activating adapters. There is a predicted coil, and like I said, at the very end, it binds SUN1, which is attached to telomeres, right? So it fits the description of an activating adapter, and long story short, it is an activating adapter. And basically, the way we showed it is, initially, we made a lot of constructs, only the N domain, N and the coil domain, a delta TM version, basically just so that, because you're trying to purify these proteins, having a transmembrane domain complicates purification, stability, and so we delete the the transmembrane domain and call it delta TM. So this is the first piece of evidence that it's looking like an activating adapter. What we used here is a technique called SEC-MOLS, so size exclusion chromatography coupled to multi-angle light scattering. So it's a technique that can be used to get the molecular weight of a complex, the stoichiometry of a complex, that doesn't depend on the shape of your complex or anything. It just depends on the mass, absolute mass of your complex. And what we showed there is, again, the molecular weight determination is the data you see in the black here. You can see that it's a homogeneous complex, whether we use the N domain or the NCC. But the instructive finding here is that the coiled-coil domain, the NCC has it, the N domain doesn't have the coil-coil domain, actually forms a dimer, just like you see for other activating adapters. So it does look and behave like an activating adapter. We then showed that it forms a complex with LIC. So we took NCC, okay, and so this is LIC alone, size exclusion profile, and secmols. So with the molecular weight, this is NCC. And then you add the two proteins together, it forms a complex with a higher molecular weight as expected. And I think the cool observation here was it forms a stoichiometry, which is a dimer of Cas5 bound to one LIC. This does not agree with what others have seen in the dynein field. They always see a 2:2. And so it either means that Cas5 is special or something else, and so we will see, or something more exciting or controversial, but that time will say. But that was an interesting finding, and we tested this always in all different ways, and we always got a 2:1 complex. All right, and then kind of the test, if you remember, I told you the way the N domain binds LIC is through this FF helix, right? The helix at the end that has an FF residue. So this is just wild type complex, just showing you the complex forms between NCC and LIC. And now you make this F to A double mutant, meaning you convert those to A's, and now the proteins do not bind with each other. They elute at different parts of the size exclusion column. They do not form a complex with higher molecular weight. Okay, so clearly Cas5 is binding LIC, and it is binding it using the same region that other activating adapters use to bind it. Okay, and then this is the final actually binding data I will show you. This is that helix sufficient to give you the interaction? Okay, that FF helix with NCC. And so we use ITC here, and we could actually get a really nice curve and get a KD, which is close to what people have seen for other activating adapters binding to the LIC helix. Okay, so I think all of this shows very strong evidence that Cas5 is a new activating adapter, but we haven't tested activation, right? We haven't tested dynein. And so what is shown here is there are two ways we can do this TURF experiment that I talked to you about. One is with purified proteins, so each dynein, dynactin activating adapter, all separately purified, expressed and purified. Or you can do what is known as a co-IP. Basically, you express all the proteins in cells, pull down using anti-flag beads if your activating adapter has a flag tag, and then elute with flag peptide, and then load it on microtubules and watch it run using TURF, right? And so we did both. In the sake of time, I'll just show you the one on the left. And basically, again, as I already showed you, if you would have a movie like this, if you see the spots, they wouldn't move without an activating adapter. You add in Cas5, if they start moving, that is basically evidence of activation, right? And so how do you analyze these data? So what you do is you generate these chymographs. And so what is a chymograph? Again, this was done in Morgan DeSantis's lab. And what you do in a chymograph is your x-axis is still the same, its distance. You're measuring how far your particle is going. And then instead of having a time-lapse movie, your time is resolved on this vertical axis. And so any vertical line basically says the spot didn't move during the duration of the movie. So that's not good news. And then any diffusive line basically means that your dynein is trying to move, but it's moving, like science always goes 2 steps forward and 3 steps backward, right? Or sometimes goes. But then the exciting finding is if you can find particles that basically really have this oblique, very, very shallow slope, meaning they're really running on microtubules, right? So that is really what we're looking for. Can we watch events like this when we add in Cas5 into a dynein prep? Or when we immunoprecipitate with Cas5 and do a TURF, right? And so what we use as a positive control is big D2, which is the best characterized dynein activating adapter. So any reviewer would want to know, did you do big D2, and how does your activator compare to that? And so clearly you can see the oblique lines here, and what you can hopefully also see is that you see the exact same oblique lines, kind of oblique lines when we add in Cas5, okay? Again, what we're looking for here is we're just measuring GFP fluorescence. So the Cache5, in addition to having a flag tag that was used to immunoprecipitate, also has GFP so that you can watch it move. Okay, and again, its velocity, meaning the rate at which it moves on these microtubules, is similar to what is known for other dynein activators. Okay, so then we switch to what we know to do in the lab, which is if you have a predicted model for your EF hand or any protein and an interaction partner, can you do mutagenesis on the surface and find out how it binds, right? Because we still haven't solved the structure of this complex. And so we did exactly that. The EF hand can be easily predicted using AlphaFold or similarity to other known EF hand activating adapters. This blue helix you see here is that LIC helix, right? The two phenylalanines are shown as these yellow hexagons. And then we basically selected a bunch of amino acids that we think are good candidates of Cache5 for binding LIC, right? And then what we did is do those, when you mutate those residues, break the interaction with LIC. And so this is a co-IP we pull down with Cache5. So this, what you're seeing is on the beads, Cache5, either wild type or mutant. And then the question is, does it also bring along dynein? And you can clearly see there are many mutants that are defective. The three most defective mutants, again, I'm not showing you the quantitation here, but when you quantify, are shown in red. Okay? And what you would expect then is if they don't bind LIC, they would also not activate dynein. And this is now showing you from a dynein kind of activation experiment from TURF that indeed those residues don't activate as well. Okay, this is all in vitro, and so it gets dry after a while, right? The question really is what's happening in the cell. So Cache5 is a meiosis-specific protein, so you wouldn't find it in any other cell, right? So we couldn't just do it in HeLa cells. But we could overexpress this protein, and if you overexpress it, it gets to the nuclear envelope, as expected. And the question is, does it also now relocalize dynein to the nuclear envelope? Okay? And so what is shown here is here we only express its partner protein, SUN1. I hope you can see it, SUN1 forms a nice ring at the nuclear envelope. And I hope you also see that dynein has nothing to do with the nuclear envelope. It's all in the cytoplasm, right? Next question, what if we now transfect in this negative control, co-transfectin, which is Cache5 delta TM? It doesn't have the transmembrane domain. If it doesn't have the transmembrane domain, it is not going to go to the nuclear envelope. SUN1 is still fine. It's at the nuclear envelope. Dynein doesn't care for the nuclear envelope, right? Nothing changed really with this negative control. Instead, if you put in flag-tagged full-length Cache5, now you see the SUN1 is a nice bright ring, the Cache5 is a nice bright ring, and most excitingly, we can actually bring dynein to the nuclear envelope. Very artificial. This is a HeLa cell, has nothing to do with meiosis, but you can at least bring dynein to the nuclear envelope. And so then the next question was those three mutants that we identified in vivo, if you express them in HeLa cells, overexpress them, they themselves will get to the nuclear envelope because they have a transmembrane domain, but they do not bring dynein there. So if you see that ring shape that you got with wild-type protein is lost when you have mutant protein, and this is just quantitation of how much signal of dynein do you have at the nuclear envelope versus in the cytoplasm, and you clearly see that take a hit when you have any of these mutations. Okay, finally, and at least for me, the hardest experiment, which is why we didn't do it in our lab, is to actually show physiological significance, which is you need to show me in meiotic cells that what you're saying is at least somewhat true, right? And so this is where Hiroki is a champion. So first of all, there is no cell-based system to study meiosis. It's just way too complicated in reality, so you can imagine it's going to be even more complicated to artificially engineer, right? So the way to study it, the way Hiroki studies it is, if you have your favorite cDNA GFP tagged construct that you want to transfect, you would inject them, electroporate them into mouse testes, stitch the testes back, come back the next day, harvest the mice, and then harvest the cells. They would still be alive for a day or two in culture, and then do all your immunofluorescence, fish, biochemistry, molecular biology with those cells. Okay, so it's a very specialized system. And so we did it, and so basically what we've done here, the cells are wild-type cells, so we didn't use a Cas5 knockout. That's step number 2. But what you see, these beautiful blue squiggles, this is showing you a cell in prophase 1. Those blue lines are synapse chromosomes. Okay, the homologs have gotten together. They're all attached to the nuclear envelope. How do you know they're attached to the nuclear envelope? To telomeres, basically, if you stay in for Cas5, this is a GFP Cas5 he electroporated in, you'll see that the green dot is basically at the end of these blue squiggles. It's at the chromosome end, which is at the nuclear envelope, right? Very nice, right? And even the mutants, the 3 mutants, now we made the numbers change because these are the mouse equivalents of those 3 residues. They're still the same residue type, just the number changed. And even those Cas5s make it just like we saw in the HeLa cells to the chromosome ends. And so this is the Cas5 panel for you. But the question is, what happens to dynein? And just for technical reasons, we've used a dynactin subunit antibody, a P150, just because we didn't have good dynein antibodies to do this. And basically what you would see is, again, the results are not as contrasting as it was in HeLa cells. So the first panel is just showing you all the Cas5s are at telomeres here. Okay, so that's good. This is now showing you how much dynein does that Cas5 bring. And one of those 3 mutants actually did show a statistically significant decrease in dynein localization, this L mutant. So we feel like, at least in vivo, it is more difficult to break this interaction. And of the 3 residues, the most important one is this leucine mutant. Time will tell when we solve a structure, the basis for all these observations. Okay, so with that, I already went 50 minutes, or almost 50 minutes, I think. I don't want to drag it further. In the first part, basically what I summarized is like a lot of work from us, ending actually with 2 cryo-EM structures that basically showed 2 regions of TPP1 important for binding 2 regions of telomerase. And the second part is our recent work on meiosis, still unpublished, which reveals this protein Cas5 as a newly identified dynein activating adapter. Okay, that moves chromosomes during meiosis. And so acknowledgements, I acknowledged everyone along the way, but those who directly contributed to these projects are their names. Most of them have now left the lab for greater things, but their names are shown in bold. I also want to thank the collaborators involved in these projects. The funding, some of it is past funding, but that led to the successful completion of this project. And last but not least, I want to thank you all for staying awake and listening. And so, yeah, please don't hesitate to ask me any questions. Thank you.
Can you hear me? So it's time for your questions. Yeah, here we go.
Organizing the telomeres in the nucleus through the membrane, which is, I think, amazing. But I was interested also in that you showed in this structure of telomere, or at least in the scheme at the beginning, that there is a RAP1 protein. And this is really interesting to me because I didn't know it has anything in common with that. But I know that, and in the light of what you were saying, I know that my field is B cells. Actually, when the B cell makes this interaction with antigen-presenting cell, it reorganizes the centrosome and the whole microtubule complex to the membrane. And through this RAP1 and dynein, organizes this whole structure of
in human cells?
Yeah, in human cells.
Okay.
So it's rather like, I think the connection is there, but probably mechanistically.
Yeah, so since they are recording this, I'll try and repeat the question. If I didn't get it right, you should tell me.
I was wanting to ask about RAP1.
Yeah. So the question is, in B cells, right, one of the sheltering proteins, so the RAP1 protein, this is one of the 6 sheltering proteins, that has a role in actually kind of reorganizing the centrosome closer to the membrane, and whether I have insights into it. I did not know of RAP1's role in doing that. I know that it is involved, for example, in yeast, for example, it is important For the process I talked to you about, it is important, I think, in meiosis. But in terms of other functions of RAP1, I do know that it has, again, maybe there is a different RAP1, I don't know. But if it's the telomeric RAP1, another function that it does have has to do with it and TRF2. So TRF2 is its binding partner. They're always thought to be together. At least TRF2 is always bound to RAP1. They are known to go to other genomic regions. So not just bind at telomeres, bind at other regions. And they are thought to have a role in in transcription somehow. But I don't think it is clearly known what that role is. This comes from chip studies, and the other thing is RAP1, it could well have this function because RAP1 is the only one of those 6 sheltering proteins where you can delete it and the mouse is absolutely fine. Okay, the other mice are all embryonically lethal. Other mice meaning the knockouts of the other 5 proteins. And so clearly RAP1 is capable of doing other things that telomeres are generally not. But I'm not aware of this specific function, so I cannot comment.
No,
Yeah.
Yeah, it's just, I didn't know about this.
Yeah.
So it's like
Yeah.
interesting that, it's like the same basic.
Yeah. But it could be telomere mediated. What I'm saying is I don't know if it is. Yeah. Yes, great question. Yes, Carol.
I'd like to ask you about TTP1. I already forgot. How does it actually stimulate telomerase? Is it DNA binding protein per se? Because I always thought that it requires sheltering.
Right. And so the part that I skip for simplicity, but is always obviously caught by experts. So that's a great question. So the answer, I can answer it to you. Okay, sorry. The question was, how does TPP1 stimulate processability in vitro? So the way we do our in vitro assays, there is a DNA primer, single strand. You can order it from your favorite company, right? 18 nucleotides. And then you have nucleotides, one of which is radio-labeled. And then you add in your telomerase, and then you plus minus TPP1, right? The protein that we purify in the lab for TPP1. And the question is, telomerase can bind a primer and extend it. We know it doesn't need TPP1, but it does that better when TPP1 is around. But telomerase binds TPP1, but Carol's question is, but is TPP1 binding DNA? Because otherwise, how does it keep the DNA, how does it keep telomerase on the DNA so that telomerase can keep synthesizing, right? So TPP1 does not bind the DNA, but TPP1 binds POT1. And all the telomerase assays I showed you are POT1 TPP1 complexes. Okay, so the POT1 is always in there. Yes?
I would like to know whether there is any update on mechanism that regulates, telomere length in cells, because I remember it's 25 years, protein counting mechanism proposed by Eric Nielsen, but is there anything new in this direction? Do we know how telomere length is regulated?
Yeah, so this is a great question again. And the question is about protein counting models. So this was something that was suggested decades ago, and which basically says, the question is, we have many chromosome ends, and telomerase, first of all, you don't have enough telomerase molecules to go to every end and extend other ends. What has been shown in several model systems is that telomerase is actually extending the shortest telomere. Because what triggers senescence, what triggers, oh my gosh, the cell has short telomeres, is the first or the second shortest telomere. So really all telomerase has to do is keep that telomere, right, above its length, and it's fine. And so the protein counting model is, how does telomere sense that? Well, maybe it just counts the number of, so in terms of proteins from my talk, sheltering molecules or cell sheltering complexes. Longer telomeres would have more of these inhibitors of telomerase, and so telomerase would not go there. Shorter telomeres will have less of these. The problem is TPP1 broke that dogma, kind of, because TPP1 is a sheltering protein, and it recruits telomerase. So longer telomeres actually have more TPP1. And so I think it has become even more difficult to answer that question of protein counting because of this. Because before this whole TPP1 business of recruitment came about, shelterin was considered an inhibitor of telomerase, right? But then, again, I was not the first person to show TPP1 recruits telomerase. That was a work from Joachim Lingner's lab, Mike Turns, and also Sunny Songyang's lab in Texas. And they showed that, no, wait a minute, you have a shelterin protein that does protection, but gives special access to telomerase. So I think the protein counting model probably has moved backwards because we really don't know. There was one study from Eros Denci's lab, but there has not been any follow-up study with this protein called TZAP1. It was a big paper in Science. And there the idea was that longer telomeres and shorter telomeres have the same number of shelterin copies. Okay? What they differ in is TZAP. So TZAP is this protein that can also bind telomeres. So the longer telomeres, that extra space is filled by TZAP. And that might be what is being counted, but I have not seen any follow-up on that. Sorry, most of my questions is I don't know, or I... but yeah.
So
yes.
over there is a question.
Oh, sorry, yes, I didn't see that.
I will, in a way, follow up on Carol's question. So, in addition to telomere, there is also alternative lengthening of telomere. So, how would you think that this, how these two processes actually potentially coexist? I mean, in cancer cells, it seems you have 15% of the cells are dependent on ALT, while the remaining are telomerase dependent. So, do you think that these two can coexist, and maybe the ALT might be the one acting on those shortest unprotected telomeres?
Okay. So I have to give more introduction for that question because again it's a specialized. So this is so I said 90% of cancers depend on telomerase. Again, depending on the review, it could be 80, 85, or 90. And so say 85% or 90% depend on telomerase. So then how do the remaining 10% of cancers still keep dividing, right? How do they solve the end replication problem? So there is no unique answer, but what people have come up with is this word called ALT, which is alternative lengthening of telomeres, meaning if it's not telomerase, it is ALT, right? And the question is, can ALT and telomerase both happen at the same time in the same cell, and then ALT would be responsible then for elongating the shortest telomere? I think from so many ways of addressing this question. So there was this huge clinical study which basically looked at, so there is now a known mechanism for how telomerase is activated. So there are mutations in the promoter of the TERT gene that are found in cancer cells, in these 90%, not all 90%, but a good fraction of them that activate telomerase, right? There was a clinical study which basically bioinformatics, which looked at ALT cancers and telomerase positive cancers. Where they saw ALT, they did not see telomerase expression. And where they saw telomerase expression, there was no evidence of ALT. For example, APBs, the kind of bodies you look for. So that has not ever been detected. What has been seen is if you knock out telomerase from mice, or I think there are also studies in human cells, and most of the cells will that's it, right? You cannot survive without telomerase. There would be a small surviving population that develops ALT, and that's the way it survives. So I do know that ALT can take over if you are deprived of telomerase, but I don't know of an instance where both are happening at the same time in the same cell.
And maybe a second follow-up question would be, how would you perhaps describe the role of the tether transcript, so another RNA with the opposite sequence, so how that is involved in regulating the telomerase?
Right. I have to again give more introduction for that question. So this but all amazing questions, and I don't have the answer to most of them. Is about TERA. So TERA is basically the telomeric sequence, like I said, for mammals. Again, it's different for different, can be different in different species. Is GGTTAG in multiple repeats, but it's DNA, right? And for the longest time, it was thought you don't transcribe telomeres because it's not a gene. But Joachim Lingner's lab and now many other labs have now discovered that there is transcription of these DNA repeats. And so what you would form is these perfect repeats of RNA. And they're called TERA, T-E-R-R-A. Telomeric RNA, it could have been simpler, but that's what it's called. And the question is, because these repeats are the same as the repeats that the template for telomerase would recognize, right? Telomerase sits on telomeric DNA and extends it. Wouldn't Terra be an inhibitor? That was an initially proposed model, but I have, at least in the recent conferences I've been and talking to people, it is not very clear Terra plays a direct role in telomerase regulation. It has important roles. So this RNA doesn't just diffuse off in the cell. It is transcribed at the chromosome end, and it stays there. And there are many studies showing that it's important, but I don't think there is a very clear connection between Terra and telomerase, even though in principle you would think there should be. Yes?
Thank you for a nice talk. I have one question regarding your TPP1 protein. You showed us nicely, and the study that was already published, that TPP1 can recruit telomerase to the telomeres. But how actually, do you have some explanation how telomerase can sense and find the very end of the telomere so that it can elongate it? Because the TPP1 cannot sit only on the very end, it can sit anywhere.
That's exactly, I mean, that kind of follows from Carol's question of the protein counting. This is a very good question. Again, I don't think anybody knows the answer to that, if I can defend myself. Is TPP1, like I said, multiple telomeric repeats, so multiple TPP1 molecules on the telomere, but telomerase acts at the very 3' end, right? You have 16 kilobases of, in a young person, not maybe Sira than me, but 16 kilobases of telomeric DNA, TPP1 throughout it, but only the very last one sitting at the end is the productive one because telomerase. So I think, has not been proven, I think it has to do with telomerase wanting to bind TPP1 where it can also engage the 3' end. So it is, if I could, I don't see a marker here, but basically imagine what are the interactions when telomerase is, okay, telomerase binds TPP1, TPP1 binds POT1, POT1 binds the single-stranded DNA. But even the single strand is very long, so you have many POT1 TPP1s, right? But another thing that telomerase binds is the 3' end of the chromosome, which is DNA, right, with its RNA template. So I think all of those things have to come together, which basically means telomerase would not very stably bind internal. It would be on and off, which kind of agrees with Jens's data, from his past papers. So there is dynamic binding. So imagine this is the telomere, this is the very end, and it can be on and off, on and off. If it binds to the very end, it just stays there, and then it extends. But it is speculation based on some single molecule studies. Yeah.
So do we have some other questions? If not, let's thank again Jayakrishnan.
Thank you.
And let me thank all of you for coming today, and I'm looking forward to meet at the next session at the Life Sciences Seminar Series. Thank you, have a great evening.